tough time, so I'm going to take my time, go through them with you. Let's help each other. We have the first one. We had the P. We had a P-shaped female crossed with a normal shaped male. And then I want to show you something here that you need to follow. Whenever we see a P-shaped chicken, we surely know that there was no capital letter that represents a dominant gene. Therefore, automatically we know that we have got two small q's. Because I said in complete dominance, once we have got one dominant gene, that gene is going to mask or hide the other one. So we only have a problem when it comes to the normal shaped one, because remember, let's go back to the example I gave you of sheep. I said a black sheep is born from two whites. What does that mean? They still have those genes, but the genes were hidden, or they were dominated by the dominant one. So when we come to this, we have to specify that this is a normal shaped but a pure breed. This is a normal shaped but a hybrid. You have to be careful about that, okay? So then this one has no problem. Once we have got this, we know this one is P-shaped because there was no dominant gene. Therefore, automatically we make a conclusion that this is a small q, which is the genotype, okay? So the genotype is a small q for this one. Coming to this one, it was specified in the question that it was a pure breed. What did I say a pure breed is? A pure breed is a homozygous. That means this one is also a homozygote or a homozygous. Homo means the same, it's a pure breed. And therefore, in this case, because it's said to be a pure breed, we shall have a capital Q and a capital Q. And therefore, these are what you call the genotypes. So that means this is a genetic makeup. It's a recessive gene and a recessive gene. Here it's a dominant and a dominant. And therefore, we need to make gametes. I'm giving you steps. These are the steps that you shall follow when you answer a question in theory. If there is a question on genetics for your paper, for your theory paper, you follow these steps. You first of all write the phenotypes. These are the phenotypes, and this will be the genotype. Remember, this determines that one, though I'm doing them in this order, but you should know this one is the one that determines the other one. And therefore, the gametes formed here, this is a female. Therefore, the ova or the eggs are going to have this. As we go deeper, you are going to see that even if we take just one, it will be representative of all of them because they're the same. Anyway, for you to follow, I am going to take both so that we keep on crossing and then we practice this performing these crosses, okay? And then coming to this one, if I take one, it's a representative of them all, but for the sake of practice, let's take both. So that means the sperm, in this case, one of the sperms is going to be like this. In fact, all of them are going to be like that. So these are the sperms from the male pure breed, and this is also the eggs from the female pure breed. Okay, so if this one crosses this one, we shall have a Q and a Q. I said it doesn't matter whether you put this one in front and this one at the back or the other way around. It doesn't matter because the makeup is going to be the same. Okay, so this one crossed with that. If this meets that one, remember this is probability, so this is what we shall have. And then if this meets that, we shall have that, or a small one before the other one. Let me do it deliberately. This and this mean one and the same thing. It doesn't matter. It doesn't matter the order. Okay? So then, out of 1, 2, 3, 4 offsprings, or out of the young ones born, we shall have the following genotypes, okay? We take the genotypes, okay? How many genotypes do we have? We only have 2. In fact, 1, sorry. We have only 1. All of them. So we say, out of 4, all of them are heterozygous. In other words, all of them are hybrids. So that's what we mean. All of them are a capital Q and a small q. So that's what we say. So this is our genotypic representation, or the genotypic result. Okay? Then coming to the phenotype. The phenotype is what we observe. Then the phenotype, I said, the presence of one capital shows that we are going to have a dominant characteristic. Therefore, we shall have a normal shape of the collar here. The same thing, because we have one Q, we shall have a normal shape, another normal shape, another normal shape. But we have a problem. The normal shape in the phenotype, we won't know whether it is normal because it has got 2 capital Qs, or just like that one, or it is a homozygote. But it doesn't matter. I shall show you how we test for that as we go along. Okay? So we say here, the phenotype is that all of them are normal shape collar. So all of them will have a normal shape collar. So all the chickens that shall be from this cross will have normal shape. Every time you take that hen and then you cross with that cock, you always have this type of offsprings. They won't change. So this is exactly what people do in farms. I think you have wondered how people manage to keep the color of cattle the same throughout the farm. This is what they do. They choose, this is how we choose the males and the female to breed. And therefore, there is going to be this, you keep to this, all the offsprings are going to be the same. And I think we are very clear on that one. Let's come to the second one. The second one, which is B, you have copied this one, I'm sure. The second one, which was B, we had the female was normal shaped, but it was specified that it was a hybrid. Okay, and then this one was also specified that it looked normal on the outside, but in the inside it was a hybrid. And remember, there's a talk that what is inside determines what is outside, and that's exactly what we're doing here. So we have got the normal shaped, but it's a hybrid. What do we mean by a hybrid? It has to have 2 different. So the genotype of the hybrid is a small q together with a capital 1, and then it's crossed with the same thing this side, a small q and a capital Q. Therefore, these are the genotype. We come to the stage of gametes. So as far as gametes are concerned, we shall have eggs this side, okay, like that, and then the sperms this side, like that. I'm giving them tails just to show the difference between that these are sperms and these are eggs. Okay, always do that. This is how you get your full marks for this type of questions. Okay, then if this comes across this one, okay, why do we have them? Remember, we have to have an N condition so that when we add one N and one N, we shall have our 2N. Remember that. Okay, so if this gene or this allele crosses with this one, we shall have this one. And what shape will this be? Remember, just one capital letter, just one capital letter represents a dominant gene. We shall have a normal. So this is normal shaped, and this is also a hybrid. And this is how people know hybrid rams. gona ge le Basotho re tumme ka digamo re rata dinku farelane mane when people go to buy rams they look for this one. If you get this one, you are very lucky because all the offspring that shall come here will always have this dominant gene. I think we are very clear. If this dominant gene is for the good fur that you are looking for, the good more hair that you are looking for, not more hair, sorry, wool, but that would be wonderful, okay? Then, if this one meets with this one, yes, this is still normal. This is still normal, but this ram is not so good. Because if it meets a sister like that, kappa another, an iwi like that, you see that small Q and the small Q will combine and they won't give us some good wool. I think you understand that. So this one is the best, and this one will be the number 2. So when they create them, they shall know that this one is a pure breed and this one is a hybrid. And we know that hybrids, for instance, you are not allowed to cross them, you are not allowed to use them twice in your flock or more than 2 times. They say, haula nehela ka yona habedi o kadimea motho e mong go tshintshana ka tse dintho tse ntse jwalo. I think you understand this based on this, okay? So that is the second possibility. Another possibility, if this one combines with that, we shall have that one and that one are the same, but on the outside, it appears normal. Why does it appear normal? I'll repeat this for you to get this. Why does it appear normal yet it has a small q? Because this one is going to mask and completely dominate this one. So this one is hidden. You won't even know it's there. Okay? This one is going to be the one expressed in the phenotype. So I have my phenotypes already, 3 of them. So we are left with this one and this one. If this one and this one cross each other, this is what you don't like for our sheep, for instance. Anyway, here it doesn't matter. We shall have a P-shaped cob. So this cob is going to be P-shaped. Why is it P-shaped? Because there was no dominant gene for the normal one. So we go to our, let's start with genetics, genotypes. So let's go for genotypic ratios. Genotypes. As far as genotypes are concerned, we have got one like this. There's none like this genotype. Okay, so there's one like that, which is said to be homozygous dominant. Or this is a pure breed. Why is it a pure breed? Because they are the same. Okay, so there's one of it, there's one like that, and then we have 2 of this, where there's a combination of different alleles, and we call that heterozygous, or we call it a hybrid, as I'm saying. And then there are 2 of them, because we have 1, 2. That is genotypically. And then the last one that you see is this combination. So there's none like this. This one is like that, 2 of this, and then 1 of that. So the ratio is that, and we call this a homozygous recessive. So be careful, there's 2 homos, 2 similar dominant, similar capital letter, and similar small letters, which we say it's homozygous recessive. I think we are clear on that. Therefore, the genotypic ratio is 1:2:1 in this type of a cross. And if you don't want to call it 1:2:1, let's talk about percentages. We say this is 25% to 50% to 25%. One thing interesting about ratio is that when you add the ratios together, you shall have 1. So it means 25 plus 50 plus 25 will give you 1, which is 100 out of 100. I think we are very clear. If you take proportions, decimal place, decimal fraction, we shall say this is 0.25 to 0.5 to 0.25. Be careful, multiple choice, they like varying these things. Sometimes they give you numbers and say there were 200 of chicken born in that farm. And then how many of those will be that? We know that 25% of 200 will be, let's start with this one, 100 of them, 100 of the chicken out of the 200 are likely to be this much, and therefore there shall be 50 of that and 50 of that. So this is in numbers, be careful. That was the ratio, this is the percentage, this is the decimal fraction, and this one is if it was real numbers, if there were 200 of them. How do you know there were 200? If we add that, and that is 150, and then that would be our 200. So we are saying out of 200 chicken born, 50 of them will be homozygous dominant. You sell these ones, if they are cocks, you get purebreds here. You buy them, you get purebreds. That means if you use this for breeding purposes, they are going to always keep that phenotype constant like that. And then 100 of them will be hybrids. They are still fine hybrids, but remember you don't use them more than twice, possibly. And then these ones, they won't sell these ones. These ones maybe they will consume because these ones we don't need them. Okay? So that's exactly what happens with these crosses that we are talking about. So my boys and girls, I think we are very clear that this is our genotypic ratio. How about phenotype? Phenotypically, there are only two phenotypes. Yeah? Remember, genotypically there might be 1, 2, 3. But phenotype, there will always be two. What we mean, it's either it's a normal one, and then the other one is it's P-shaped. So those are the only two phenotypes that we shall observe. And then I shall use this part of the board after we remove, let's extract the numbers that we see here. Out of 4, we have got 1, 2, 3 that appear normal. Remember I said this one and this one and this one, on the outside they appear the same just because of this. One capital Q. That capital Q is the key role player here, and therefore this homo, once we have got a dominant gene which is present here, is also present there, so we shall have 3 and then we shall have 1 like that. So our ratio is 3:1. Okay? In a farm of 200 chicks, how will these results be? Remember, this and this go together. This is from the same cross, and therefore we are saying, as far as numbers are concerned, this 1 and 2 in the phenotype join together. And therefore, we have got the normal one, the normal shaped ones will be 150, and then these ones will be, we have 50 of them, which is the P-shaped. Okay, so the normal is 150 and the P-shaped is 50. If you check your numbers very well, if you apply your maths, you see that the ratio is going to be 3:1. If you express it in percentage, it will be 150 of 200 multiplied by 100%. Surely you shall have your 75%. And this one, that's a 50 out of 200 of 100%. We apply our maths, we shall have got our 25%. You add 25 to this, you shall get your 100%. I think we are very clear. So that is the solution for that one. So the genotypic ratio is 1:2:1, or 25%, 50%, 25%, or numbers 50, 100, that one. Coming to this, phenotypic ratio 150 is to 50 if we talk in terms of numbers, and the ratio is 3:1, and then percentage wise 75% is to 25%, or we can say this is 0.75, this is 0.25. So any of this will be fine. Thank you so much for this one. Now let's do the last one. The last one was a hybrid. So it means from that 2, remember the ratio was 1:2:1. Let's pick one of those two, meaning we pick the normal one, the normal shaped female, but it is a hybrid, meaning it is like that. Okay? It is a hybrid, and then you cross it with a P-shaped. Remember, once we have a P-shaped, we know there was an absence of a capital Q. So it will be like that. It means we are crossing that and that one. Okay? So what do we say? Then, in this case, we shall have 2 types of eggs, and then we shall have 2 types of sperms. I said if I take one, it's fine. If they are similar, we can take one. But for practice, let's take both. Then that one crosses with that one. This is normal. Okay. This one crosses this one. This is QQ. This is also normal. Remember, these are the genotypes. These are the gametes. And then we shall get to our zygotes. And then this one and this one, we shall have that. This is P shape. Okay, this one and this one is another P shape. All right. So here we have a different situation, different scenario. There are 2 phenotypes and 2 genotypes. Let's start with the genotypes. So the genotypes, we only have 2. It's either a heterozygous, which is a hybrid. There is also a homozygous recessive, which is a pure-breed. This is a pure-breed, right? Those are the 2, unless you see any other. I can only see those 2. I can only see a capital Q and a small q, and then we have got small q. Those are the only the genotypes that we have. And therefore, there are 2 of this. It means there will be 2 of that, and then there will also be 2 of that. So it simply means the ratio here is said to be 1:1. 2:2 divided, simplified, will give us 1:1. What we mean, we have 50% of that, 50% of that. Okay? If it was a farm of 400 chicken, and then you cross them like that, it means about 200 of them will be like that, and then another 200 will be like that, 50%, 50%. And how about the phenotype? Let's end with the phenotype. So as far as the phenotypes are concerned, there are 2 phenotypes, the normal to the P-shaped. Okay, so the normal and the P-shaped. And how many of them are normal? There are 2 of them, and then there's 2 of them that side 2. 1, 2, 1, 2. And then the ratio is also 1:1. So in this case, be careful. I want to end by saying this. When both the genotypic and the phenotypic ratios are 1:1, like in this case, we call this type of a cross, this cross is known as a test cross, or we call it a back cross. Okay, what is a back cross or a test cross? This is when we cross a hybrid with a recessive, homozygous recessive. And then the ratio is going to be 1:1, 50%, 50% for both genotype and phenotype. We call this a test cross. When we perform a test cross, when you have bought a ram, let's say you want to buy a ram for merino ram, if it produces very nice wool, kapa dominant wool re re ke T. And then re batla ho tseba hore ramo ya rona, is it like that or like that? Why can't we know from the appearance? Because the presence of just one T will confuse us. Ha re na tseba hore na ke dominant tse pedi kapa ke dominant le recessive. Remember, we can't know from looking at this unless you perform a test. Re nke nku fela e sefoka fela e boya bo bobe re tlo e crossa le yona. Okay? If all offsprings from that cross come with good wool, it means ramo ya rona ke, because all of them will have good wool. I think we understand that. But if it was like that, re crossa le yena, what will happen? Some of our lambs will have good wool, some of them will have bad wool, 50%, 50%. Then re tseba hore ha ra reka ramo e ntle, re rekile ena hybrid ena. Ke ane re itseng o neela ka yona habedi, then you go and get another one, you go and buy another one. That's why you see farmers will have more than one, because they are trying to vary this, because there are chances you won't know. You won't know when you go to buy whether it's like that or like that. I think it makes sense. And boys and girls, that is about monohybrid crosses, complete dominance. The next one that I shall do will be incomplete dominance. This is where we talk about blood types. How come you are blood type A, blood type AB, blood type B, blood type O? Remember, we did talk about that. Thank you so much.